Hello there, and welcome to Smith and Sheridan, a podcast on the science and business of biotechnology, presented by me, Cormac Sheridan.
And me, Andy Smith. Hello, everyone. Hello, Cormac. How are you?
I'm doing good. How are you, Andy?
I'm well, thanks.
You felt the need to wade into the very dense thicket that is the subject of cancer vaccines this week. And they are obviously a very mainstream topic in oncology, therapeutics development, I suppose you could say. And it's a field with a lot more failure than success, including recently Gritstone Bio.
That's why I tipped my toe into the water of cancer vaccines and now realised it's the Mariana Trench or something. It's the analogy fixes. But yeah.
Okay, so we're down from the thicket and we're now into
an ocean,
a very deep trench.
Yeah.
And of course, subsequent to the Gritstone Bio news, which we can go into further, there was another development in the field. Andal Therapeutics reported some very promising data, and they got a big bump on the share price. It went up by 280% or something in one day from an admittedly modest enough base for their pancreatic cancer vaccine. And if you can get any movement in pancreatic cancer, that would really address major unmet need. So one can understand the investor exuberance, and it is based on survival data, not based on response data. So it's kind of looks pretty solid to me. They've reported a median overall survival of 28.8 months versus 12.5 months for those in the control group of a phase 3 trial. It is an interim analysis, but that's a very big survival benefit, it seems, more than double. And at 24 months, the survival rate for those on the CAN 2409, as the name of the therapeutic, it was 71.4% for those who received the vaccine versus 16.7% of those in the control group. Out to 36 months, the survival benefit did detract somewhat. Not all patients were surviving beyond 24 months because you had estimated survival was 47.6% in the 2409 group versus 16.7% in the control group. But if that holds up, and they'll obviously need to do it in a larger study, but that's very interesting. And what's also interesting to me is that it's kind of oldish technology. It's a replication defective adenovirus, which delivers a herpes simplex virus thymidine kinase gene directly to the tumor. And it induces, therefore, and when you get concomitant delivery of the prodrug, you basically get lysis of the cells that take up the adenovirus and express the viral thymidine kinase. And that's the killing of the cells. And then you get, obviously, an immune response from the lysed contents of the tumor cells. It's very, very reminiscent of a previously approved therapy, also viral space, which was tended to be called an oncolytic virus. This is Amgen's Imlygic, which was approved all the way back 2015 now at this point in melanoma. That was a modified herpes simplex 1 virus, replicates in tumor cells. It also produces GM-CSF, the immunostimulatory cytokine. And same idea that you get virus-mediated lysis of the infected cancer cells. And that was always a very modest product. It didn't have a very pronounced efficacy. You couldn't do it systemically. It had to be delivered intratumorally. And therefore, it was only to lesions on the skin and the lymph nodes or cancer lesions that were basically biopsyable, I think is the shorthand way of putting it, where you can get a needle into it, you know.
And as you say, Cormac, the last time I looked at Amgen's results, very modest, the sales?
Very modest, deeply, yeah. They were broken out, the revenue numbers. It's in the other products category.
Yeah, oh, right, yeah, where products go to have a quiet life,
So it's in the sort of retirement home at the end of their product tables. And I doubt if it does more than 100 million a year at the very most. I don't have the number.
Which is a paradox of cancer vaccines, right? Because we're into a number of approved entities, lots of clinical studies, and they should already be bigger than they are, shouldn't they?
Well, I'm a firm believer in things being as they are and accepting them as they are.
Right.
That's probably why I'm not a successful biotech executive. I'm just a commentator, because I noticed that people think that we should be further down the field. And I've heard any number of pitches from people, we're developing this, that, and the other, it's all great. And
me too.
Cure the dogs, cure the rats, whatever. And yet, the amount of approvals we have, the amount of slam dunk clinical data that we have is very, very limited. But it's growing. It is growing. I mean, you could say that, it's, to paraphrase the former presidential candidate, it's the immune system, stupid. The interactions between cancer and our immune system, we're only barely beginning to scratch the surface of that in terms of understanding. I'm sure that will vary as well considerably from patient to patient. And trying, therefore, to intervene in that very dynamic kind of a scenario, which is also fast-moving, to achieve some sort of clinically useful outcome, it's a very, very complex task. And
we've talked about things like receptor redundancy and pathway crosstalk before, how medicines we think should work and actually don't. The topic of the discussion today is cancer vaccines, and vaccines have done wonderful things for humanity, yet it's not in cancer vaccines, it's everything else. And you can sort of see, we were delivered from the pandemic by mRNA and to some smaller extent protein and even AAV vaccines, and that worked.
No, adenoviral vaccines, just for the record, on AAV.
Is it? Not AAV, okay.
Chimp adenovirus was the
AstraZeneca one. Yeah. So it works for vaccines, but as you say, and as you've intimated, tumors are a bit more wily than that. That they mutate every 5 minutes. They chop and change the antigens that they express, and they move around. They don't move around, but they move their antigenic profile such that an antibody response on its own is not enough. But as we were talking about, I would have thought, so for influenza, for most of our vaccines, including COVID-19, an antibody response was enough. But fair enough, because this is self we're talking about. These are our own cells, mutated admittedly, that we're talking about, that we need to address. So yes, I think it's a bit late in the day for me to say this. I'm sure everyone else recognizes it, that you need cell-mediated immunity. You need activated T cells to get in there and attack by whatever means you prompt that. Is it going to be by a virus? Is it going to be by BCG, which we know works for bladder cancer or helps with bladder cancer? Or is it going to be mRNA? My worry is that it's taken this long for us to get this far, that suggests that it's still not going to be easy. We should have done it by now.
Yeah. Obviously, when one thinks of cancer vaccines, our generation, you think of Provenge. And the amount of hype attaching to that product, and it did, I think, kind of cast a pall over the area for some time. And concomitant with its kind of commercial failure and its inability really to sort of deliver at scale for patients was the rise then of the checkpoint inhibitors, which didn't have the same complexities. They were antibody-based therapies and eminently scalable, didn't require any awkward business in terms of isolating people's white blood cells or dendritic cells and fiddling around with them. I mean, Provenge was approved in 2010. I think Yervoy wasn't. Was that 2011 or so? The first of the checkpoint inhibitors.
But even then, that is not a PD-L1 or PD-1 inhibitor. No. So they never really took off until they happened.
CTLA-4, yeah. But it was a little bit later for the PD-1s, But Provenge, I mean, it did appear to offer about a 4-month survival benefit if you look at the FDA label. But I think they really struggled to scale it because just the actual extraction of the cells and their modification, now they weren't genetically engineered, but they were just kind of exposed to various activating agents.
What do you call something, Cormac, or what's the phrase for something that's meant to do something, or a politician that's meant to give a speech about something, and it completely goes the other way and ends their career, if you like, or ends the party? Because Provenge did do a good job at pushing back the whole cancer vaccine field for a long time. When you look at the data, and I think I've commented on this to you, that you look at the data, extended overall survival, which is great. Even if it's 3-4 months, if it's $60,000 or something for 3-4 months, I'm sure that means a lot to a lot of people. But
it was about $93,000, which at the time was considered astronomical. And now it's I hate to say the phrase chump change, but it's
compared to gene therapies.
Yeah, even the most recently approved cell therapy from Iovance, Tagvi, which you could argue, I don't know whether you call it a cancer vaccine or not, probably it's called cellular therapy.
Mm-hmm.
They're charging $500,000 for that, roughly speaking.
Yeah.
Absolutely extraordinary.
And that's the other positive thing about Provenge. But the negative things about it was it extended overall survival, but it didn't extend progression-free survival. And you think the problems, late-stage metastatic prostate cancer have is they have to be on opiates because they have lots of bone mets and they're in pain, they can't move. So you pay now $90,000 to live longer with all the pain you wouldn't have had you not had that, which strikes me as a bit paradoxical, but...
And I'm sure you're not paying $90,000 now. I'm sure it's a multiple of that these days.
Yeah. But the one good thing that Provenge did was that it was a tailored therapy. It was specific to each patient. And that opened the door for logistical issues that have been met or are being met by products like the CAR-Ts. And we even have CAR-Ts for solid tumors now. So.
And what's also very interesting to me is that we have a sort of a Provenge Mark II kind of shaping up. And it's not the same product, but DCL-VAX-L from Northwest Biotherapeutics for glioblastoma. There's been a marketing authorization application filed in the UK in December. So they described it as activated autologous dendritic cells exposed to cancer neoantigens. So it's slightly different from Provenge, and it's in development for patients who have operable glioblastoma. So the idea being that you take a sample of the tumor and you expose their dendritic cells to that sample in order to hopefully, quote unquote, train the immune system on the tumor so that you put it back in the cells and Bob's your uncle, you get a response. And they showed they've had a Provenge-like survival benefit in a phase 3 trial. Median OS of 19.3 months versus 16.5 months for matched external controls. I don't know. You harumphed when I mentioned it.
I know. I mean, Northwest Bio was an AIM stock years ago and then transitioned to NASDAQ. And its DCVax cancer vaccine has, like Northwest Bio, has been years in the failing.
Yes.
And there have been many commentators who tried to get to the bottom of its clinical data and not come up with anything positive. And Northwest Bio is like one of the... There are certain sectors of our industry that attract people, and I'm not saying Northwest Bio is one of these, but biotech certainly is one of these where you could, if you'd gone back 100 or 200 years, you would see the same sort of people touring America selling snake oil. Biotech has this, and unfortunately, because of what's gone on before, cancer vaccines have that moniker, but potentially with the mRNA vaccines coming along, and the vaccine you already mentioned for pancreatic cancer, which is a very difficult ask. I mean, people don't get diagnosed with pancreatic cancer. It's symptomatic until the very late stages. That's why the survival is so poor. Perhaps the end may be in sight, or an end may be in sight, but we've certainly not covered as a sector, cancer vaccines, we've certainly not covered ourselves in glory over the years.
No, we haven't. But just on Northwest Bio, they're certainly not the darlings of the stock market, but they have a healthy kind of market cap now. But I mean, these phase 3 data, they were reported in JAMA Oncology. So you're talking, substantial peer review. You are talking about people from very, very well-recognized cancer treatment centers across the UK and the US, and maybe one or two other countries as well. I don't know what more do they have to do to get the market
in the US. America is the world's largest pharmaceutical market by value.
Yes.
If you had a safe and efficacious drug, why on earth wouldn't you, as most companies do, why on earth wouldn't you launch it there first? Why would you try and go to the MHRA, not even the MEA anymore? Why would you go to a tin pot country like the UK?
Oh, don't be so hard on yourself.
I'm just asking the questions, Cormac, rather than giving the answers.
Yeah, I think that's a fair point. And that, I do think they're in phase 3 and they do have ambitions around the US, but maybe more momentum will get behind it.
And the person at the FDA who reviews vaccines in a press articles recently said they're poised to review articles.
Yeah.
I think it was very good of him to say that, but he didn't say we're poised to approve them. He said we're poised to review them.
Which takes us to, and I think the piece you're referring to in Fierce Biotech, it was Moderna Merck vaccine, which has, to me, solid momentum behind it. The data look interesting. As far as I know, the data are reported in press release and maybe conference presentations. I don't think necessarily they've appeared in a peer-reviewed paper. Am I right?
I don't know.
But they've really done really well with this.
Well, Merck are an oncology powerhouse, right? You'd think they know their onions when it comes to oncology products.
Yes. And it's referring to mRNA 4157, which is a neoantigen-based mRNA vaccine. So in other words, the vaccine encodes, I think, 20 neoantigens derived from the genome of each patient's tumor. And at 3-year follow-up in their phase 2b trial, so when you gave people after surgery tissue, and the cancer was removed as much as it could be. So people then received the mRNA plus Keytruda, Merck's blockbuster PD-1 inhibitor. The combination reduced the risk of recurrence or death by 49% versus Keytruda monotherapy alone. And it also reduced the risk of distant metastasis or death by 62% versus Keytruda alone. That's, to me, for people who've become refractory to PD-1 inhibitor therapy, that looks very promising, doesn't it?
It does. And the whole neoantigen thing, for me, is an interesting one, right? Because I've seen a number of companies over the years who've isolated a range of neoantigens and put them into clinical trials and come up with nothing in terms of a benefit.
Gritstone Bio being a case in point.
Yeah, and Immunicum in Stockholm. But the thing is, neoantigens are new antigens to the immune system, right? It does make me wonder why. I mean, not everyone dies of cancer, right? Unfortunately, we die of heart disease or we walk in front of buses and things. So it makes me wonder, on a population basis, are there people out there who raise a cell-mediated response to their own neoantigens when they had a solid tumor cancer and resolved it? And are cancer vaccines doing essentially the same thing?
I would imagine so. I think our immune system is constantly monitoring for any irregularities. And for whatever reasons, some people develop cancer, other people never develop cancer. And it's not to say, that there weren't subclinical events occurring that the immune system was able to take care of without you even noticing.
But in the same way as APOA1 Milano was discovered, its role in cardiovascular protective health, and familial mutations have been responsible for a few important drug discoveries. It makes me wonder why companies like 23andMe aren't out there looking at patients or families who've got incredibly low incidence of cancer and wondering what is it that their immune system is doing that we want everyone's immune system to do.
And I think that work has been happening. I mean, I think also, for example, quite a number of years ago, the US NIH or NCI was doing a study looking at people who had maybe extraordinarily effective responses to clinically evident cancers. So people responded really well to therapy. The real outliers in a positive sense.
Now we're getting through. So I worked in the CRO once, right? And the CRO, certainly when they're doing oncology studies, the clinicians and the clinical monitors talk in hushed tones about the hyper-responsive patients. Because if you can find one of those and stick them into the active arm, you're on to a winner.
Oh, right. Well, this wasn't anything as nefarious as that. It was purely an attempt to uncover new biology. But I would just add the caveat that interrogating something as complex as an individual's immune system and its response either to a clinically evident cancer or its theoretical response to cancers that are not evident, that's not trivial. And you can basically generate reams of data, and then you'll have to use some sort of AI system to try to help you interpret it.
Which is what Moderna and Merck are doing, sort of.
Yeah, they're doing it obviously in a way that's manageable because they're trying to basically make the best picks in terms of the antigens. And maybe that's what was Gritstone Bio's undoing. Because if you have an ability to select neoantigens that will result in immunogenicity, that there will be an immune response against them that will have clinical consequences, that selection process is extremely important. And I mean, one of the great kind of pioneers of this area was Ugur Sahin of BioNTech, and he had done quite a lot of the running and publishing papers in Nature, et cetera, about doing this. And yet it's interesting to me that Moderna, their great rivals in terms of mRNA vaccines and therapeutics, seem to be a little bit ahead of them. And the Genentech, the early deal between BioNTech and Genentech, signed, well, practically almost a decade ago now, this stage, not quite a decade ago, but certainly more than 5 years ago. It's not delivered much yet, but at the same time, they have reported very promising data in pancreatic cancer. Mm-hmm. It was Nature, actually, I think it was a cover story in Nature in 2023. And after 18 months of follow-up in a phase 1 trial, those who did respond to the vaccine, they hadn't reached median recurrence-free survival. Basically, the Kaplan-Meier curve was a huge, big separation, because those who didn't respond, kind of an internal control on the trial, the non-responders, they had median recurrence-free survival of 13.4 months. So that's going to be very interesting to see how that matures. And I don't...
And as you say, Cormac, I mean, for years, in decades, a decade or so before the pandemic, BioNTech were there with cancer vaccines or mRNA cancer vaccines, aligned with a diagnostic for each product, and had some useful collaborations. But we go back to the fact that this is not easy. Biology doesn't play ball, and cancer doesn't even want to play at all. So
I think we're in a process, I wouldn't be dismissive of the whole field at all. I think we are in a very typical phase in biotechnology innovation that various approaches are being tried. Some are old and not very sexy, but they may be delivering, or the wheels might come off the wagon. We might uncover insights into what makes clinical trials flawed or not. There is innovation in terms of the mRNA, and I think Moderna and BioNTech, Moderna are more advanced, but I think they're going to deliver. But it mightn't be the case either that you're going to deliver every single time, that different cancers have different immunological properties, or not properties, characteristics maybe. And it's also very interesting that different cancers have different propensities to produce neoantigen diversity and bulk. So I think, though, the clinical trials that are ongoing are going to deliver a wealth of data. And I think certainly the kind of momentum that Merck and Moderna have with their particular construct, which their lead indication is melanoma, but they're also non-small cell lung cancer, and they plan to move into other indications this year. I think that's going to be an incredibly important program. And right now, I think it's the sort of the one that's kind of, to me anyway, is going to be the sort of the poster child, if you like, for mRNA-based neoantigen cancer vaccines.
I, on the other hand, Cormac, are wondering whether we've seen the poster child yet for even cancer vaccines, even mRNA vaccines. Because, and the reason why I say that is, you know, when CAR-Ts started happening and they showed efficacy, but the patients were really, really ill with cytokine release system. You knew that the hematologic tumor burden was being addressed because the patients had CRS. Now, are we seeing that with solid tumors? Are we likely to see that with solid tumors? I mean, you're likely to, but less so. We're almost doing ourselves out of a marker here, but quite rightly so. Because in the adjuvant setting, where you remove most of the tumor surgically before you treat the patient, and most of that tumor is gone, so less likely to have a tumor lysis syndrome effect. And the theoretical aspect we were talking about earlier, where there must be some people who address their own, raise an immune response, cell-mediated immune response against their own cancers. At that time, when their very highly primed immune system detects it, the tumors must be so small that you don't get a tumor response, or you just feel a bit under the weather or something. So I don't know what markers need to be there, but a marker of, toxicity would be horrible for the patients, obviously, but a good one to see.
Yeah, I think that's a very interesting point, and I do not know the answer to that question. I don't know whether it's in the nature of the type of response you're getting that you do or don't get these. I actually just generally don't know. I'm just kind of having a quick look at the Nature paper, which the BioNTech people and their colleagues in various clinical cancer centers that did this work. We're only talking a small number of patients. This is like 16 patients. I'm just seeing whether or not there was any cytokine release. And there's nothing.
Was that melanoma, or was that?
This is pancreatic cancer.
Oh, right, yeah. Because pancreatic cancer is diagnosed so late, you might see it. But then if it's in the adjuvant setting and most of the tumor is removed before you start treating, then you won't get it.
Basically, the protocol involved treatment with atezo lizumab as well as with the RNA. So typically they are given as combinations, and that's just it's a PD-L1 inhibitor.
Right. Yeah.
And basically, from what I'm seeing, they treated some patients with the PD-L1 inhibitor, and none of them had grade 3 or higher adverse events.
Mm-hmm.
So yeah, in the phase 1 trial that BioNTech and their colleagues reported in Nature last year, 1 out of 16 patients, that's 6%, who received the mRNA vaccine, had grade 3 adverse events or higher. Actually, just grade 3 adverse events. That was fever and hypertension.
Mm-hmm.
All 16 of them had grade 1 or grade 2 adverse events. And when you then co-administer the two agents, the PD-L1 inhibitor and the mRNA vaccine, that doesn't matter actually. That's just an efficacy thing. So basically, from a safety perspective, it doesn't look like, in these patients at least, that results in CAR T type of responses. Now, don't forget too, of course, that the CAR Ts expand after administration.
Yeah, they self-expand, and also the tumor burden is distributed throughout the body. So you'd expect, rather than one single site, you expect for a tumor or even metastases. So yeah, the potential for a systemic cytokine release system obviously feels like it's much higher.
Yeah. Because certainly the BioNTech people were saying that the vaccines did stimulate a T cell response in pancreatic cancer. But to me, it looks like it must have been localized or not sufficiently intense, that it didn't cause cytokine release syndrome.
But the good thing is, Cormac, that when we do, or if we do, if that turns out to be a marker of efficacy in cancer vaccines, from the CAR Ts, we know how to treat it these days. So
we do. Yeah. So I think we're kind of at this I hesitate to say that we're at this halfway point. I think we're at a point where we have some agents that are active. We have multiple modalities in play, and that's appropriate. That's always the case. And whether or not you want to regard oncolytic viruses as cancer vaccines or not, to me, the boundary between them is kind of blurred. But that's a whole other day's kind of discussion too. We have some autologous cellular immunotherapies, and we also have some sort of engineered viral therapies. And now we have the mRNAs, as well as obviously Moderna and BioNTech. You have CureVac, which is earlier stage, as it always has been. Yeah. And you also have obviously Gritstone, which obviously hasn't delivered to the satisfaction of the market.
Or you don't have, I don't think you have Gritstone. Let me summarize what you've been saying, Cormac, is we have lots of new technologies, lots of new vapes, but no cigar yet in cancer vaccines.
No cigar, I accept that. But I would also say watch the space closely.
Oh, yeah. And I mean, and I, like you, I'm sure, hope for the day where those patients we hypothesized about that cure their own cancers, where everyone could be one of those with the right treatment.
Yeah. I mean, there is so much unknown about our immune systems, and so much that's probably misunderstood about our immune systems as well. But I think when you get there, when you get good clinical data and you can understand what's happening, you can then start to maybe make some mechanistic inferences. Because a lot of the time, people think it works this way, but the actual fine details of it might suggest otherwise. But that's just the nature of clinical science. It's just fiendishly complicated. And I salute those who actually engage with it and generate new knowledge in the field. Long may it continue. Until the next time, Andy, thank you very much.
Cheers, Cormac. See you, everyone.